Okay, thank you, Jackie, for that very nice introduction. So it's a real honor to be here and to be able to tell you about our work on how electrons and protons move together to enable chemistry and biology. So I want to start out just by describing electrons and protons. So a hydrogen atom, we probably all know, has one electron and one proton, and the proton is around 2,000 times heavier than the electron. So historically, the proton has usually been described as a point charge here, like this blue in the middle here. And then the electron is treated quantum mechanically as a wave function. So in other words, as a delocalized probability density. You can see that by this red electron cloud, and you can see there's a darker red in the middle here. And that means higher probability of the electron being there, and that's because the proton has a positive charge of +1, and the electron has a negative charge of -1, and opposite charges attract, and so that means that the electron is going to want to spend more time there in the middle near the proton. Now, when two atoms get together, they can form a chemical bond, and then the electrons rearrange so that they can get more electron density here in the middle between the two nuclei. And chemical reactions involve the movement of electrons because they involve the breaking and the formation of bonds. And here's a very simple chemical reaction. You just start out with a water molecule, H2O, and you have two bonds, right, between the two OHs. And you add a proton and you form another bond now, another OH bond. And so clearly the electrons had to rearrange themselves to form that new chemical bond. But there's another kind of reaction involving electrons, and that is electron transfer, or ET, from one site to another. So here you see two iron atoms in water, and the irons are shown by these brown spheres here. And here on the left, you have an extra electron, this mesh around that iron, and then that electron can transfer to this other iron, the one on the right, and so now that electron is now localized on the right side. So that's an electron transfer reaction from one site to another. And it turns out electrons can move over very long distances because they're so light. All right, and Jackie already mentioned Rudy Marcus, who developed a theory for electron transfer that allows us to predict the rates. And he did this back in the 1950s, and then he won a Nobel Prize in 1992. But what about the protons? It turns out that chemical reactions can also involve the movement of protons. So we can also have proton transfer. So we call electron transfer ET, and proton transfer would be PT. So you can have PT, proton transfer, from one site to another. So here we go with another water molecule. The oxygen is red, the two white spheres are protons. And you can see there's a proton moving between two water molecules. And on either side, it forms a bond, a chemical bond with the oxygen. So it's just going back and forth. This is a proton transfer reaction. Now, the proton is going to move a much shorter distance than the electron because it's 2,000 times heavier, right? So it can't move those lengths. But still, it's very important when the proton moves like this. Now, I already mentioned that usually the proton is treated classically, like a point charge, this blue in the middle here, and that the electrons are always treated quantum mechanically. But it turns out that sometimes the protons also need to be treated quantum mechanically. They also need to be treated as a wave function or as a probability density instead of just a point charge. And so here you see a quantum proton. Instead of a point charge, it's a delocalized proton. Not as delocalized as the electron, right, because it's still 2,000 times heavier, but it is still delocalized. It's not actually a point charge. Now, it turns out that electrons and protons can also tunnel through barriers. So if you think of this black curve here, that black curve is, say, train tracks. There's no train on it. But what we're going to do is we're going to take a ball, like a billiard ball or something, and we're going to just drop it so that it's moving back and forth on this left side for the classical particle. And it's just going to, if there's no friction, right, it's just going to move back and forth. Right? It's never going to get over the barrier because it doesn't have enough energy to get over the barrier. So it's just going to stay on the left side. It's stuck there when it's a classical particle. But if we decide we're going to treat that particle quantum mechanically, so we're going to describe it with a wave function or a probability density, right? In this case, it's one dimension, so it's this blue curve. That's the probability of where the proton is. And you can see from this picture on the right that then for the quantum proton, it can actually move from one well to the other without actually going over the barrier. It doesn't have enough energy to get over the barrier, but yet it's going back and forth, right? It's on the left now, and it's going to go on to the right. And that is quantum mechanical tunneling. It's going through the barrier instead of over it. So it turns out that electrons are always assumed to be able to tunnel, right? They're so light. So all of our electronics, your cell phones, which are hopefully turned off, but those electrons are always tunneling wherever they go. But it turns out that protons can also tunnel. They're just going to tunnel over shorter distances because they're so much heavier than the electrons. But we still need to think about the fact that the protons are tunneling as well as the electrons. Now, I already mentioned that a proton is a positive charge of +1, the electron is a negative charge of -1, and opposite charges attract. So it turns out that many times the proton and the electron will move together. And that leads to this phenomenon that's called Proton-Coupled electron transfer, or PCET. Now, it turns out that PCET is important throughout biology and chemistry. So here are two examples. There's on the left, we have photosynthesis. On the right, we have an enzyme that's essential for DNA synthesis. And what we see is, if you look closely, you don't worry about all the details, they're obviously very complicated, but you see electrons and protons transferring in different places and in different directions in these complex processes. Now, PCET is a much newer field than the field of electron transfer. I mentioned Rudy Marcus started back in the 1950s with that. So it's a newer field, but it's still become very active because people realize now that PCET is ubiquitous. And I just list a few names of some of the people who performed the pioneering experimental studies of PCET. And you can see Dan Nocera, who's I think here in the audience, is one of the people who was really the leader in this field. So what my group does is we develop theories to be able to describe PCET. So our goal is to understand where the electrons and protons are going and how fast they get there. And we really want to be able to predict how to change and control the pathways and the rates of these PCET reactions. So to do this, we needed to derive a lot of mathematical equations and expressions. We're treating the electrons and the protons quantum mechanically to include all those effects of delocalization and tunneling. And then we get expressions, say, for the rate constant. That's what's shown here. I'm not going to go into the detail and explain any of that. But my point is that we have these expressions, and then we need to use computational methods, simulations and calculations, to be able to calculate the input quantities for this rate constant expression. And some of these methods are very standard in the field of chemistry. So, for example, if you want to describe molecules, you solve the quantum mechanical equation, you obtain an electronic wave function, and then you can calculate the geometries, the relative energies, and the vibrations. And I show a molecule here. So each one of these spheres is an atom. And the different colors correspond to different types of atoms. So you can see the hydrogens are the small white spheres. There's, again, the oxygens in red. And what this is a structure of a molecule, and you see it moving, and that's just a vibration that it does at room temperature. It's just doing this all the time. Now, if we have large biological systems, we can't treat everything quantum mechanically, right? It's too expensive. So people have come up with methods more approximate. For example, molecular mechanical force fields, where each atom is represented as a sphere, and then each configuration of atoms has an energy associated with it. So, for example, a chemical bond would be described by a spring between two spheres. Steric effects would be described by, if you think of two billiard balls knocking into each other and bouncing back. And electrostatic interactions, again, I already mentioned opposite charges attract, and then like charges will repel. So all of that goes into this big expression to describe big molecules. Now, if we want to know how these molecules are moving, we can do molecular dynamics. We just solve Newton's equations of motion. Force equals mass times acceleration, and we get the forces from the energy. So if you think back to high school physics, right, if someone throws a baseball, you know the initial position and the initial velocity, then you can say where that baseball is going to be at any time, just assuming the only force on it is the gravitational force. Now, we can do the same thing with a protein, which has many atoms in it, where each atom is sort of treated like a baseball, where we're just solving force equals mass times acceleration. Its force is going to be more complicated than gravity, but yet we can still do this numerical integration here. And here's an example of an actual protein moving. So here, all we're doing is doing force equals mass times acceleration on all the different atoms. And we don't even show the individual atoms. This is such a huge system. We just show these ribbon structures, and you can see this thing vibrating. Again, that's what it's doing at room temperature. But so far, when I've talked about these computational methods, there's no quantum mechanics, right? Everything is being treated classically. But I already told you how important quantum mechanics is for electrons and protons to delocalize. If we want to break or form chemical bonds, we need to describe the electrons quantum mechanically. If we want to describe a hydrogen tunneling, we need to treat that proton quantum mechanically. So my group has developed methods to go beyond the standard methods and incorporate this kind of hydrogen tunneling in these kinds of simulations. So this green mesh here, that is the proton, the nucleus. And what you just saw was that it tunneled and it's going to start again. And you can see it starts out basically as a sphere, but then it's going to delocalize. It becomes like the shape of a peanut, right? And it starts to delocalize between the two wells. And there it goes, and now it tunneled. And so that's tunneling in an enzyme, and I'll point out that this was a collaboration with Steve Benkovic, who's here in the room from some time ago. So now we have the methods and the theories to be able to study these systems. I want to talk about two examples where we've used these kinds of methods to learn something useful. And one is, the first example has been inspired by PCET and photosynthesis. So plants convert energy from sunlight into chemical energy, into chemical bonds. They're storing the energy in chemical bonds, and that involves PCET. So what you see over here is the ET for electron transfer, the PT for proton transfer. There are a lot of things going on. So the idea of this project was to figure out, can we follow nature's example but design molecular systems that do PCET? So we'll use an electrode now, so a slab of metal, to take an electron from a molecule and get a proton to transfer. And we just apply a voltage to get that, or a potential to get that proton to transfer, and then we can maybe get more protons to transfer. Here there are 2 protons transferring. So the idea is to assemble what's called a proton wire. Now we all know about electrical wires, where electrons are going through all of our electrical wiring in our houses. Well, these are proton wires where we get protons to move along a wire. And the reason that we care about this is because many solar energy devices, which convert sunlight to chemical bonds, they require the movement of protons over long distances. So this was a collaboration with Tom and Anna Moore, and they've been designing artificial photosynthetic systems. And what they did is they said, let's look at this photosystem 2 on the previous slide, and let's just represent this red part of it by a molecule, this molecule called BIP. And so what they're doing is designing molecular systems to mimic PCET and photosynthesis. And our calculations have been helping with that. And the idea is, in this case, you remove an electron and you can get 1 proton to transfer here. But we want to transport protons over long distances, although each proton can only move a short distance, right, compared to an electron because it's so heavy. But the idea is to have multiple proton transfer reactions to create a proton wire. And here's a picture of that. We have our electrode, we apply a potential, we take an electron, and we can get 1 proton to transfer or 2, and so forth. And what we did is in this collaboration, it was a feedback between experiment and theory. We could design molecules on the computer before they even made them in the lab, and then they test them out. Or sometimes they made them in the lab, and then we helped them interpret their experimental data or helped them improve the molecules. And so in this case, you can see we have a series of molecules that we got 2, 3, and 4 protons to transfer. So what we're doing here is we're removing an electron, and then we can get, it's easier to look over here on the left, you can get 4 protons to transfer from one side of the molecule to the other. Each proton's only moving a short distance, but the net transport of protons is over 16 angstroms. And then the question is, well, are all these protons transferring exactly at the same time? Now, it turns out our experimental collaborators, they're doing electrochemistry. That's too slow of a timescale to figure out, to really answer that question. But on the computer, we can answer that question. What we can do is we can take the molecule, just remove an electron, and we do molecular dynamics. Force equals mass times acceleration. And that's what we're doing here. You can see they're all moving around. That these 2 white spheres here, those are the protons that are transferring. And what we find is they're not happening at exactly the same time. Even though our experimental collaborators said all they see is all of the protons either in their initial places or in their final places, nothing in between. What we're seeing here, you can see there's a delay between these 2 proton transfers. That the first one occurs about 100 femtoseconds after electron transfer, and the second one occurs about 600 femtoseconds later. So basically, you can see there's a delay between it. There's a delay, and there it goes. Right, so PCET is asynchronous. They couldn't see that experimentally with electrochemistry, but we can see it on the computer. And it turns out there are experimental techniques that can look at this very fast time scale. And so we've now put this prediction out into the literature, and the hope is that some of these people who look at faster timescale processes will be able to then answer the question, do they actually see this asynchronicity? So in this project, what I showed is by just removing 1 electron with an electrode, we can transport protons over long distances. So there's a net transport of these protons from this side of the molecule all the way to this side of the molecule through a whole series of individual proton transfer reactions. And this can be important to designing solar energy devices, and we all know that solar energy, right, is very important as an alternative energy resource, right, that's sustainable. So the second example is an enzyme called ribonucleotide reductase. So ribonucleotide reductase is often called RNR. This is an enzyme that's essential to DNA synthesis and repair in all living organisms. So it turns out that it's a target for cancer chemotherapy. So inhibitors of this enzyme are currently being used, right, in cancer treatments. So we want to understand how it works. Now you can see it's a very complicated system. It's, you know, there's this big mess on the left here. And we're very fortunate that Kathy Drennan and Joanne Stubbe have used cryo-electron microscopy to solve the structure of this protein. In other words, they now know where all of the atoms are. Now there's some uncertainty, but still, we have a starting point here. And for computational, for simulations, we need to have that starting point. We need a structure of the protein before we can start simulating. And so now that we have this gives us an ability to step forward, to move forward. And what's really interesting about this structure is it shows a PCET pathway that's spanning these two subunits, this red and the blue here. These are just two pieces of protein that are interacting. And they have 6 PCET reactions spanning 32 angstroms. And it looks, I know, very complicated over here, but basically proteins are made up of residues, and the residues here are labeled with these capital letters, so Y, for example, over here. And what this enzyme is doing is it needs to transfer an unpaired electron over 32 angstroms. So usually electrons come in pairs, right? You have many pairs of 2 electrons. But sometimes there's an unpaired electron called a radical. And if, for example, if you have an odd number of electrons, you're always going to have one unpaired, right? So this radical is shown by that red dot there. And so the idea is to transport that radical, that unpaired electron, clear across to get to here, because you need to get that to that other side to be able to do the chemistry that's required to synthesize DNA. And we want to understand how that happens. So it turns out it happens by transferring an electron between residues. And remember, electrons are much lighter than protons. They can transfer over larger distances. So this electron is going clear across from this residue to this, to here, and it's hopping along until it gets to the end. And it's coupled to proton transfer. So these are actually all PCET reactions. And so to understand this, we did molecular dynamics simulations again, and we focused first on this one residue here. This residue, it's called a tyrosine, and it needs to flip out here to do electron transfer with its left neighbor, and then flip back here to do its proton transfer or electron transfer with its right neighbor. And so this kind of a flipping motion was seen in our molecular dynamics simulations. Now I'll say in the structure, these two residues, they were stacked. There are 2 rings, and they're stacked, right like this in the structure. But when we start doing molecular dynamics simulations, we see that this flipping starts occurring. And you can see that with a cartoon there, but I think it's more interesting to look at the real atomic level simulation here. This is that residue I'm talking about, that it's flipping back and forth because it needs to do PCET with its left neighbor and PCET with its right neighbor. Right? So it needs to go back and forth. We get this level of atomic, resolution from a simulation. Now, I will point out that Dan Nocera and Joanne Stubbe, they had experiments that actually showed for a slightly modified enzyme, they were able to show the same sort of evidence of the same sort of flipping. We can do it on the original enzyme, but also we can see this atomic level detail that they can't see in their experiments. But again, I want to emphasize this importance of the feedback between experiment and theory. We're always going back and forth, and we're learning from each other. And I think that's very important. If we can't just do theory, our theory always has to be validated by experiments. Now, then we wanted to look at how the proton is transferring between these 2 stacked residues that I mentioned, right? You have a proton going between these 2 oxygens. Everybody just assumed it went directly between the 2. But when we did molecular dynamics simulations, we found that another residue, this one up here, comes in and actually participates. So you can see from this movie, this other residue is sort of shepherding the proton, that white sphere, across. That's something they didn't know experimentally. We identified this residue as being important. And then Dan Nocera and Joanne Stubbe actually changed that residue into something else, and the enzyme died. It no longer worked. So this is an example of how simulations can identify key residues, and that's going to be very important in designing proteins, modified proteins for, say, drugs, drug use or for inhibitors, better inhibitors. This is a way to actually stop the reaction by changing this residue into something else. Now, I will say we've done additional calculations showing that hydrogen tunneling is also important in this enzyme as well. So to tell you what I've sort of summarized what I've been telling you about, the coupling of electrons and protons, right, and of their movement is essential throughout chemistry and biology. It's important to understand how to control where the electrons and protons go and how fast they get there. And so we've been developing theories and computational methods to be able to treat the electrons and the protons quantum mechanically in these kinds of calculations so that we can describe proton-coupled electron transfer in an accurate way. And our theories and simulations have been providing insights into very complex systems. There's an example here about designing proton wires that can be important for solar energy devices, and also elucidating enzymes, like this one here that's essential for DNA synthesis, so this could be important for drug design. And then I want to acknowledge the people who did the work. This is my current group at Princeton. There's some former group members. These people are the ones who actually did the work I talked about today, and our experimental collaborators, and also the places that fund this research, and I'd be happy to answer any questions. Thank you.
Super, super. All right, questions for Sharon? Yes, please.
Scientists who try and explore the universe make the statement that it's just chemistry and biology, therefore would be the same anywhere. Is there anything with sufficient time resolution or some other resolution that can prove that the chemistry that you've been demonstrating here is really happening elsewhere? I mean, we've got spectroscopy, but that's really broadband compared to the resolution that would be needed to verify that chemistry really is the chemistry somewhere else.
And you're talking about somewhere else, like in the universe, not on Earth?
Yeah.
Okay, yeah. I mean, clearly some of these reactions, for example, require oxygen. So you need to be in an environment that requires oxygen. But I think, there are certainly in atmosphere, chemical reactions that there are computational chemists, theorists who are studying those kinds of reactions out in the universe. And so I would say it does depend on the conditions. It will depend on the temperature. And again, sometimes I think, obviously the temperature is quite different on some of these other planets. The degree of oxygen, which I already mentioned. So I do think that the chemistry depends on the conditions. So I don't think you can say that all chemical reactions will occur on all planets. But I think some chemistry is likely to be occurring on, everywhere. It just might be slightly different types of chemical reactions because of the different conditions. But certainly you can simulate under these other conditions, different pressures, temperatures, and so forth, with or without oxygen. So I think that is something that can be done on the computer, and that is being done. And there are many computational astrophysicists, for example, who are addressing those sorts of problems.
Yes, in the back.
So that was a terrific talk, and I was wondering whether you look into the effects of replacing the protons with deuterons. That must affect the tunneling substantially. This is Will Happer from Princeton.
Yes, a great question. Yes, that is the kinetic isotope effect. So we calculate these things with hydrogen and with deuterium, and then you calculate the ratio of the rate constants of hydrogen over deuterium, and you can get kinetic isotope effects. What's really interesting, so yes, we care very much about that. I didn't put that in this talk, but that is something that experimentalists measure and that we calculate. And it turns out, for example, there is an enzyme called soybean lipoxygenase that has a kinetic isotope effect of about 80 at room temperature, and you can make some mutations, you can change some residues and get that kinetic isotope effect to go up to 700. These are experiments that were performed by Judith Klinman at UC Berkeley, and we've also studied these theoretically, and our theory has been able to explain that at all. Of course, that is a hallmark of hydrogen tunneling, because they're replacing one hydrogen with deuterium, right? The difference in mass is only a factor of 2. Yet the rate goes down by 800 or 700 or 80 or 700, right? That's clearly hydrogen tunneling, and that's actually further emphasis that you really need to include hydrogen tunneling in your theories and in your computations to be able to describe that kind of effect. It really has to do with overlaps between proton vibrational wave functions. That's how you can get those huge effects, by just changing one hydrogen to a deuterium. So yes, and it's a really wonderful tool to be able to understand the mechanisms of these complicated systems, is by looking at those isotope effects, but not completely straightforward to interpret, I'd say. So you really do need theories and models to balance, to go with the experiments to be able to interpret these kinds of results.
Yes, please, and please say who you are.
How in the world do you make your simulations run fast enough to get the results you show? These are very complicated molecules.
Yeah, that's a good question too. So, I mean, yes, most of these aren't happening on the femtosecond timescale, and when you treat things quantum mechanically, it gets more expensive. So you're absolutely right. There are methods, enhanced sampling methods that allow you to sample the regions of interest by putting restraints on certain atoms. So you want to see a chemical reaction to occur. You would restrain certain, like say the hydrogen to be in the middle, or to be along the pathway. If the hydrogen moves along here, you would do a series of windows where you're doing simulations and you're restraining the hydrogen to be in each one of these positions. And then there are statistical methods to weight these in a way that you basically get the results for the actual real system without the restraints. So that's a type of enhanced sampling method. So there's a lot underlying this. There are a lot of, and of course, my group is just one of many, many that are studying these. I would say that also I should emphasize that other groups have also worked on including quantum mechanics into these kinds of simulations. And many, many other groups have developed enhanced sampling methods that we can just use because they took the time to develop them. And so you do have to worry about these things and develop computational algorithms. So that's all part of the field of theoretical chemistry, yes.
Well, I think given the time, and with that, let's thank Sharon once again.